Hello everybody, my name is Mabel Gonzalez. I'm a chemistry student of Bogota, Colombia from the Universidad de los Andes. I have to blend both biology and chemistry in order to study the chemical ecology of one of the most coolest animals in nature, which are poison frogs. So I was very fortunate because I was born in Colombia, one of the most biodiverse countries in the world. So let's take a look to the numbers. We are the number one in biodiversity of orchids and birds, and we are the number two in biodiversity of amphibians, plants, and other organisms. Many of the areas of Colombia are covered of thick and sparse jungle, as you can see in this picture taken from the airplane when I was arriving to Quibdó, the capital city of Chocó, one of the most important states in Colombia in terms of biodiversity. So the Dendrobatidae family, which is the family of poison frogs that I was working on, they are only distributed in the neotropics from Central and South America, and they are accumulating these toxic compounds in the skin. So the skin is actually the sample that we are collecting in the jungle and analyzing using GCMS. This family of frogs are very famous for the different colors that they have, which are called aposematism, and they advertise the predators of the toxicity with this specific colorful coloration. The first people that discovered the toxic compounds in the frogs were the Embera Indians from Colombia, which actually were using the poison of the frog for envenomation of the darts, and then with blowguns they were hunting monkeys or other prey just for consumption. Right now, the indigenous people are not using the poison frogs anymore, but they actually still know how to build and construct the blowguns and the darts, as you can see in this picture of a current Embera indigenous Indian person. So thanks to John Daly, who led the frog alkaloid program at NIH in 1963, trying to figure out which are the structure of the Indias were actually using in the darts. Now we know more than 800 different alkaloids from different amphibians in the world. And so unique is the chemistry discovered in the back of these frogs that we have specific families for these specific compounds. And they are only found in frogs and the arthropods that they are eating, because that's another surprise. These frogs are not producing or synthesizing these chemical compounds, they are actually sequestering from the diet. So that's another thing that we are still trying to understand, because we know that they eat it, accumulate it, and transport it to specific glands in the skin, but we actually don't know how this is happening in the frogs, which is really interesting. Some of the families that we can find in the skin of the frogs are pumilatoxin, stronicotoxin, gephyrotoxins, batrachotoxins, and these are pretty unique of only these two sources, frogs and the arthropods that they are actually eating. What do we know is that in the back of these frogs there are a great power for drug discovery, as it happens with epibatidine, one of the alkaloids that were isolated many years ago, and it has 200-fold more power than morphine for being a good analgesic. So the nature, as you can see, provides endless inspiration for us to discover new biologically active compounds that may, for example, become new drugs. And this is one of the motivations for studying these specific animals. So now I'm interested in studying the interindividual variation of the chemical profiles of the frogs, because when they were discovered and the first structures were proposed, the differences between specimens were hidden. And I'm interested in knowing how big are the difference between different individuals and how this is related with different ecological variables that I have in my metadata. So for completing this task, I've been able to annotate known and unknown compounds. There are several analytical challenges that I have to overcome. One of them is the unique chemistry of the frogs, because this is very interesting, but it's also a problem because I lack of standards that we're gonna be helping me to make a full annotation, a level 1 annotation. So another problem that I have is the reference data, because it's very sparse and it was built in 2005. So for example, for this specific GC data, I don't have retention indexes that were helping me to annotate the compounds. And the last problem that I have is that the amount of sample that I have, because many of these alkaloids are in trace levels, so they are really difficult even to detect in some of the equipments. That's the reason why it's better to use mass spec instead of NMR for targeting this specific unique chemistry of the frogs. But now we have molecular networking for GCMS data, which can help us exploring the chemistry, and I'm going to show you an example of how these networks can be useful in a situation like we have, where we have limited reference information for the compounds that we care about. So now I have submitted my first job in the GNPS GCMS platform. Now I can generate a network of the chemistry that have been detected by GCMS on the skin of these frogs. In this network, the color scale is showing the cosine score. So red means a poor match and green means a good match. And the cosine score is a value related to the reliability of the notation when my experimental fragmentation patterns were compared against the libraries that we have. In this case, the comparison were performed against Wiley and NIST. So when I take a look closer to one of the clusters of one specific compound that I'm really interested on is the pumilatoxin 251D. This is not the only one, but one of the few demonstrated cases of a metabolite compound by the frogs where they were hydroxylated to a more toxic version known as allopumilatoxin 267A. So, as you can see, the node for this pumilatoxin is green, which means that the annotation is pretty much reliable. But when I take a look to the cluster, I can see that this pumilatoxin is connected to a totally different compound, as you can see here, which is an ester. And after some years of experience analyzing the typical compounds found in the derivative frogs, we are pretty sure that this annotation of a highly carboxylated ester, as you can see for the many hydroxyls, is probably wrong. Even if I look through the other possible matches in GNPS, I can see that none of them make any sense. In the network, the spectra are clustered based on the similarity of the spectra. So even if the compound is identified incorrectly, I will be networked with the relative compounds that have similar fragmentation patterns. So we're expecting that these two specific compounds, the pumilatoxin and the other one, have similar fragmentation patterns. Therefore, using the pumilatoxin, which is a reliable annotation and could be my starting point, I can explore the annotation of this compound that I presume to be incorrectly annotated. So the first thing that I'm going to do is overlap the experimental mass spectrum and the library query for this compound. As you can see, the overlapping is not such good. The overlapping is just using the fragment of 166 for doing the match with this specific ester. Now, when we compare the mass spectra of the pumilatoxin 251D and this unknown node, we can see that there are many fragments shared between the two compounds. For example, is the 250, the 208, which is the loss of a 3 carbon of the side chain, and there are also fragments, even the 166, that in this case is the bigger fragment. For the pumilatoxin, these fragments can be attributed to different structural fragments that you can see in this picture. For the other compound, we see that the fragment 70 is absent, which probably means that the compound could be an isomer of pumilatoxin 251D, but lacking the pyrrolidine moiety, that it was the one related with the 70 fragment. Therefore, we need to take a look at the amphibian family alkaloids. And we see that there is a related family to pumilatoxins called homopumilatoxins. And the only difference is that this family of alkaloids are compounds of 2 members of 6 rings instead of 5 and 6 members as it happens in pomylotoxins. Additionally, there is another family of alkaloids when you see a demethylation in the carbon 9 in the alkaloid. And this family is known as 9-desmethyl homopomylotoxins. Having this in mind, now instead of the most likely incorrect annotation that it was proposed initially as an ester, we are proposing a new putative annotation of an alkaloid. This alkaloid appears to be new, not previously reported in Daly's library, and is found in trace amount in some of the species that we are analyzing in the network. We have called it 9-desmethyl homopomylotoxin 251 EE. Why EE? Because we are following the structure that was built by Daly for assigning the code names in the library that he were building in 2005. Such annotation propagation analysis is now possible using molecular networking, as you can see with this specific example using the unique chemistry of the poison frogs. So now we are very excited to continue using GNPS GCMS platform for annotating the compounds and proposing putative annotations for possibly new compounds. The chemistry of these frogs is very rich, and most likely many new compounds are still waiting for being discovered. This actually is not surprising, as one of the studies has been proven in Argentinian toads, where when they were comparing single skins, they were also able to find new compounds. So we are expecting to find any more. Now, thanks to GNPS, we are gonna be able also to share all these files and talk to other researchers we are working in the same area, trying to find parameters for performing the annotations and being able to discover more compounds of the unique chemistry that frogs and toads all over the world are producing in the skin.